Thanks all for being here. It's 1:30, so we're going to get started. It's my pleasure to introduce Dr. Alex Marand. Alex got his BS at Virginia Tech in biochemistry, followed by a PhD at the University of Wisconsin-Madison in plant breeding and plant genetics, before postdocing at UGA, where he was not only an NSF postdoctoral fellow, but also received a K99 pathway to independence. So very well decorated in his postdoc. Something really remarkable about Alex and that makes him really fun to talk science with is that he's got really great breadth as a scholar, and I think we're going to see some of this today. He has that background in biochemistry, but he also knows about field-based plant breeding, population development. He's worked on questions in evolution and adaptation, and he's a really good molecular biologist and computational biologist to boot. He's only 9 months into his appointment as an assistant professor at University of Michigan, yet he's got a CV that reads like that of a tenured professor, which is fantastic. Not only a tenured professor, I would even argue a full professor. But yeah, so he's been very productive. He's doing really exciting creative science that's at the leading edge of plant gene regulation, and he's gonna tell us all about that today. So give it over to you, Alex.
All right. Yeah, thanks, Joe. That's a really nice intro. I'm so thrilled to be here and to meet with you all. I've had some really exciting talks with several of you this morning, and I sort of hope that we can keep the discussion alive, because there's just nothing more exciting than engaging science, and I hope to deliver that to you. So most of what I'm going to talk about today was work that I did during my postdoc in Bob Schmitz's group. As Joe mentioned, I did start my lab recently, about 9 months ago, but if you want to talk about some of those things, I'm more than happy to discuss some of the more recent science that's happening with my group. But just want to give a quick outline, just a little bit of background about my motivations, what kind of science I'm interested in and the questions we're trying to solve. A little bit about some of the first data sets that we generated. That was the main focus of my postdoc, and then some of the more current work that's nearly published, and some things that are on bioRxiv that I'd like to share with you all, because I think it's of interest to a broader group. So I think most people, when you ask them, who they work on any sort of molecular or biological system, will tell you that they're trying to understand the relationship between genotype and phenotype. And I nicely fit into that group of biologists as well, I'd like to think. So there's a whole host of within interest-specific phenotypic variation that to me is quite beautiful. But yet we still don't really have a full comprehensive understanding of sort of how all these morphological and colorimetric diversity arises, particularly within a species, and sort of what is controlling that at the molecular scale. But one thing that we sort of have come to appreciate is that the majority of trait-associated genetic variation, that is genetic variance that is statistically associated with phenotype, tends to be located in non-coding regions of the genome, by as much as up to 95% in plant species. So when I think about non-coding regions, I think about things like cis-regulatory elements, or things that can control gene expression and lead to a phenotypic change. So for those of you who don't think about cis-regulatory elements, maybe as much as I do, you can think of them as just sort of the landing place for transcription factors. So more or less, they encode binding sites for sequence-specific transcription factors that have some sort of quantitative or qualitative influence on gene expression. Right, so you get binding of, this is just an overgeneralized picture of a transcription factor binding to its cognate CRE and turning on gene expression. So there's different flavors of cis-regulatory elements. I think the one that's probably most well-known and best understood are promoter regions, or core promoter regions, typically located next to the genes that they regulate, although that's not always the case. Becoming better understood are enhancer elements, which can be quite distal to the genes they regulate, or even within the genes that they regulate. These are obviously much trickier to identify based off of just pure in silico methods, because of their sort of independence to the genes that they regulate. And then of course, the last would be these insulator or boundary type elements, which are far less characterized in plant species in particular, that more or less shield the effects of regulatory elements to within some sort of spatial domain. So in this example, an enhancer and silencer and promoters can only act on the genes that are within this boundary. So much less is understood about those, and I won't talk about them any more than that, but just know that they do exist. All right, so some of the best evidence, actually, this is probably my favorite example of how regulatory diversity arises or gives rise to phenotypic diversity. And so this is probably the best well-known example of a insertion of the hopscotch element upstream of a gene called TB1. And so what TB1 does is it suppresses lateral branch formation. And in teosinte, you have an active, you have a inactive or lowly expressed TB1 gene, in which case results in lots of axillary branches that gives rise. Modern maize, you have highly expressed TB1, which suppresses the formation of those branches. And if we delete TB1, we can revert back to the ancestral. So this is just one example of the single cis-regulatory variant that results in a very dramatic phenotypic difference morphologically, right? And it was also sort of the target of selection by humans that made it more agronomically feasible to plant higher diversity or higher density of plants. All right, so that's just one example of a cis-regulatory element, but how do we identify these things using sort of like biochemical approaches? There's many different methods, but I think the most popular one and best or most widely used is the ATAC-seq, which stands for Assay for Transposase Accessible Chromatin. And it's a quite elegant experiment, and the rationale for that is it's just very simple. And it basically takes advantage of a highly active TN5 transposase that's loaded with sequencing adapters. And because TN5 has a preference for accessible chromatin as opposed to closed chromatin, it'll insert these sequencing adapters specifically into accessible regions of the genome. And because these universal sequencing adapters are well known, you can design primers against them and amplify adjacent regions that have these adapters inserted. So more or less, after we create a library using this tagmentation-based approach, we can amplify the library using the universal adapters and then basically map the reads and look for regions that show enrichment for accessible chromatin. And just this in terms of terminology, we typically refer to these as accessible chromatin regions. So regions that show enrichment of accessible chromatin fragments. All right. So some of the things that, my major motivation at the time was, what is the role? Can we define a role for cis-regulatory elements in specifying, maintaining, and then further facilitating the differentiation of plant cell types? And at the time, we didn't have access to some of the methods I'll tell you about in a second. So this was really a big unknown, and there's only a handful of examples that really shows that in a molecular way. And then the last question that I've sort of become interested in is how do variants within cis-regulatory elements that disrupt sort of the maintenance or specification of different cell types and identities result in traits that you can see with your eyes, right? That could have agronomic use. All right, and so the challenge at the time, again, was, we had a lack of methods, but another thing that people who don't work on plants sort of don't understand is that we don't have these pure, stable cell lines that are specific to our homogeneous cell identities that allows us to profile cell type specific processes. Because as soon as we digest the tissue or disrupt the tissue, the cells will become dedifferentiated and totipotent after a few days. So it's really challenging to maintain plants' cell lines. And so we can't use that as a way to understand cell type specific processes. But we can use known marker genes, right? So we can clone some genes and have them express or use the promoters of known cell type specific genes and have them express GFP in specific cell types. And then we can use things like FACS to sort them out, right? Or manual isolation methods. But sort of the problem with these is that you have to do a separate transformation for each independent cell type, right? And doing laser capture microscopy or any sort of manual isolation of different types of cells is very laborious. So what we needed was a method that could do this sort of in a high throughput way. So sort of the summation of those challenges has really limited understanding sort of chromatin biology in plants at sort of only the tissue or bulk level, with very few cell type resolved data sets. All right, so enter single cell methods. And this is really what I cut my teeth on as a postdoc. But the way it works is we can isolate nuclei, right? So this method allows us to sort of overcome those issues that I just described. But we can isolate nuclei using simply staining nuclei with DAPI or any other intercalating fluorescent chemical, or we can isolate nuclei using detergent-based methods, which just involves washing nuclei and purifying them from a heterogeneous sample. We'll tagment the nuclei with TN5, again, using universal adapters, but then the change occurs. We can load the difference between regular ATAC-seq and single cell ATAC-seq is this step here, where we load the tagmented nuclei onto a microfluidics chip, which generates an emulsion of droplets that contain a single nucleus and a single barcoded bead. And each of these beads contains a distinct oligo 16-mer, which gets attached to all the accessible regions from a single nucleus. Right? So in this example, you can think of, this purple bead contains millions and millions of copies of a single 16-mer, and they all get attached to all the accessible regions specific to this nucleus. And so then when we break the emulsion and amplify the libraries, we can more or less identify that all the accessible regions that contain the same barcode came from the same nucleus. Principle of single cell methods. All right, so we apply this to a sort of panel of 6 different maze organs. In total, we got about 55,000 nuclei. And one of the things that you can do is you can plot the relationships between cells, and more or less, the closer 2 dots are to each other, the more similar their chromatin accessibility profiles are genome-wide. And the big takeaway is that we can more or less separate cells out by their tissue of origin. And that's kind of the expected. Different tissues have different molecular profiles. But when we zoom in on these larger clusters, we still see additional sort of layers of heterogeneity that when we look at known marker genes kind of shows that this is our cell type level resolution. So just as an example, there's 2 genes in bundle sheath cells that are specifically expressed. If we look at sort of the distribution of chromatin accessibility among cells for those 2 marker genes, you see a co-localization of signal that indicates that the same cells both show enrichment for the same marker genes that's specific to that cell type. Right, same thing for mesophyll cells. It's a different population of cells that shows specific enrichment for 2 marker genes that are only found in mesophyll. We can also look at sort of the aggregate of all the cells as more of like a traditional way to think about epigenomics data or bulk type data is we can aggregate all the cells that correspond to a different cluster. And so in this case, we see these are 2 genes that are marks of xylem identity. So kind of all in all, we got about 52 different types of cells from this particular data set. And so one of the things that we want to know is that can we use this data to come up with new markers, right? Because if we're relying on all these old markers in future experiments, we just want to be able to use something more comprehensive. So we were wondering if we could use the markers that we would predict would be highly specific if they show sort of the expected patterns. And so we teamed up with Andrea Galavotti's lab at Rutgers with Dr. Zhongli and Chen, who did the RNA in situ for us. We had 5 total marker genes that we have some predicted pattern of expression. I'm just showing you one of them. It's GRF transcription factor 36, and we predicted that it would be specifically expressed in the inflorescence meristem and spikelet meristem tissues of the developing tassel. And when they did the RNA in situ, this is exactly what we see. So this is just one example out of several that we did, but it gave us confidence that like the predictions that we're making are actually biologically. Okay, and because I'm really interested in the relationship between chromatin and transcription and how variation within regulatory elements can lead to differences in transcription, we have to actually profile transcription, right? And the transcriptome. And so we generated some single nucleus RNA-seq data and integrated those data sets using an approach that's used to find sort of commonalities between different modalities. And then we can, of course, annotate those using known marker genes, and then look at the correspondence between the accessibility of a gene versus its expression. And a lot of those things, sort of line up well as to what you might expect. So that kind of allowed us to ask a couple more questions, sort of more biologically relevant than just, can we integrate these different types of data? So I'm just going to jump over here real quick. And this is kind of a summary of looking at the cell type diversity in chromatin accessibility profiles and transcription. So the columns here correspond to different cell types, and the rows correspond to different genes. And what you should see is that by and large, they overlap pretty well. There's about a 60% correlation between accessibility of a gene and its expression profile. But there's this other portion that doesn't overlap nicely. We call them the accessible yet silenced set of genes. And that to me was very curious, is like, why do you have these constitutively accessible genes or highly accessible genes if they're not being transcribed? Because this was kind of a commonality idea that had been propagated in the field, is that, well, if it's open, it must be expressed, right? There was no indication that it would be anything else at the time. And so I was interested to know, okay, well, what could these be? So we pulled out those genes specifically, and when we looked at some other chromatin modifications, we found that they were highly enriched with K27 trimethylation, which is a sort of developmental signal of gene silencing. And then we took, despite being totally overlaid with this highly silenced mark, they still maintain accessibility in their promoters, right? It's a lower level of accessibility, but they're still accessible, sort of on aggregate. And so when we took the accessible regions for the accessible yet silenced genes that have this K27, we found that they were enriched for PREs. And PREs are polycomb response elements that recruit the PRC2 components, which is the enzyme that's responsible for K27. So it was kind of like it was a cool sort of finding for me, because it confirmed that even things that are inducing silencing still have to be able to bind chromatin and bind DNA in order to mediate their effects. All right. One of the other cool things I thought, actually, I'd never published this result, but I was always really excited by it, was doing a sort of in silico ChIP-seq. And for those of you who work on plants or are interested in the interaction of DNA binding proteins with different parts of the genome, you know that getting ChIP-seq to work for transcription factors is really, really hard and very expensive, if not impossible. There are things like DAP-seq now that you can do, but this is even cheaper, right? Because the data already exists. So the way this works is by having an integration, right? We can impute transcriptome profiles onto the cells that we use for ATAC-seq and vice versa, so that every cell in this data set has both a transcriptome and an accessibility profile based off cells that it looks very similar to. And then what we can do is we can look at across all these cells, right? You can do this in a cell type specific way or across all the cells of the tissue. We can look at the expression level of any given transcription factor and the level of accessibility at a specific locus, right? And if I do this for all the accessible regions in the genome, I might find, in this case, I found 6,500 that have a statistically significant positive association between expression of the transcription factor and the level of accessibility, right? And so this gives us an indication of the regions that are associated with the expression of this gene. Statistical, right? But if I take these regions and I look at what sequences are enriched de novo, we actually find another BZIP, right? So this is just an example of one, the Maze BZIP61, and we identify a de novo motif that looks very similar to a published BZIP motif. So I think there's, the ability to profile many different types of transcription factor binding sites or associated transcription factor binding sites in different contexts that's based off of expression level evidence. All right. So sort of the last thing I want to say about this particular study was that, and this is what really motivated some of the next questions, my postdoc and also in my lab currently, is that when we look at genetic variation within accessible regions, we generally see a very strong like purifying selection signature. Very little amounts of genetic diversity are occurring within regulatory regions, just generally. But when they do occur within regulatory regions, they tend to be strongly associated with trait variation, right? And if you're a cell type specific accessible regions, right, you're a regulatory region that has a very strong developmental component, you tend to have an even more outsized effect on traits, organismal traits. All right, so that kind of brings me into sort of the current fold of what I'm working on. And so the idea was, well, can we leverage the single cell approach to understand genetic variation through the lens of regulatory diversity? And so what that means is we took a bunch of different maize genotypes and isolated nuclei and did a taxonomy. But because all of these genotypes that we are focused on have been previously sequenced, we can go back, right, for each nucleus and say, what genotype do you look like? So it's a very quick way to profile many, many genotypes from just a single reaction, because we can go back and in silico assign genotype identities based off known SNP pattern. If there are questions, you should feel free to raise your hand if you need clarification. Okay. So that gives us a way to like very quickly, yeah, yeah.
So you're pulling
Nuclei, yeah, from different
all
genotypes.
those universes.
Mhm. Yeah.
And then you are assigning them based on this.
Yep, because we have all the reads from a single barcode from one nucleus. We can compare its SNP distribution or its alleles compared to the 282 HapMap, right?
Okay, I have another question then.
Yeah.
So with the 4 to 27, what was that's a pretty big range. What's the?
Well, because I did this over many batches.
Okay, I
Yeah.
got
It was a practical thing. So I would grow them
Got
in large batches. And then the very first time I did it, I grew out the full 282 and I took what I could, ended up being 172. We're still had enough tissue for me to sample after 10 days. Yeah, but then the 4 to 27 was, I was supplementing to make up cell counts later. Okay, over many batches.
And these are seedlings.
Yeah, seedlings.
Okay,
Yes. All right, so in total, after making 40 libraries with the help of Dr. Xuan Zhang and Bob's lab, we ended up with just under 700,000 nuclei that we could very reliably assign a genotype to. So not only do we need a lot of reads per cell in order to say this is a cell and this is some sort of background noise, but we also have to be very confident in the genotype label of that individual nucleus, which can be a challenge if you have too many genotypes in your pool that things can kind of start to look similar to each other. So anyways, we needed very rigorous amounts of filtering in order to make reasonable conclusions sort of downstream. Just want to point out that, in this sort of scenario, we removed the genotype effects and we're purely trying to illustrate and evaluate the cellular diversity that's present in all these different samples. We have all the marker genes and things like that we use to evaluate data. So I think the first sort of question for me was, okay, now that we can define regulatory regions for each of these different genotypes, right? I'm just ignoring the cell type specific information for now. How much conservation do we have in regulatory regions just across the species, within the species? And so what I'm showing you here is a sort of non-mutually exclusive plot where we have the fraction or the number of genotypes that have a very low alignment to an ACR or accessible region that we identified in the reference genome versus the number of genotypes where we have a good representation of that sequence. So this is the sequence level conservation of regulatory. And so in total, we had about 80-ish thousand accessible regions, non-coding specifically. We exclude anything that maps to an exon. Just purely focusing on non-coding DNA. And it's about 70% of regulatory regions are highly conserved. And that's also supported by across species conservation. So looking at levels of conservation in grasses, using phylo-P as sort of a proxy for that. And just to give you an idea of what this looks like physically in a browser, you have regions that are highly conserved The sequence exists in all the genotypes. Whereas you get some level of conservation where the sequence will disappear or will have appeared more recently in some of the individuals. And so to me, those are potentially very interesting and sources of phenotypic innovation. All right, so just kind of digging into those regions a little bit more. Conserved regions tend to not be very associated with transposable elements, whereas if you're a dispensable regulatory region that doesn't show high levels of conservation, you get upwards of 25% of those are mapping to EPs. And sort of another way that you can evaluate how important a region is not only its conservation rate, but what kind of activity does it have. And so we mapped enhancer activity via StarSeq onto all these regions and then asked, okay, well, how conserved are you compared to how active you are? So typically the things that show higher levels of enhancer activity tend to be the most conserved. And then we can also, because we've genotyped every individual in this population, we can also ask the level of conservation of different transcription factor binding sites and which transcription factor binding sites may contribute more towards conservation than others, right? So the things that tend to not be conserved very often are often transcription factors that are more involved in environmental responses, stimuli to the environment, local things associated with local adaptation, as opposed to transcription factors that are more sort of encoded or encoding development and less sort of responsive to the environment. So that all made sense as sort of a way for us to benchmark what we're seeing. But I think this is kind of the most obvious use of this data is, instead of, you may have heard of expression qualitative trait loci, where we're looking for genetic variants that may affect gene expression. We can do the same thing with attack data and try to identify single variants that affect accessibility, right? So here I've clustered these genotypes based off of the genotype of some variants, right? So if you're homozygous for Ts, tend to have very low levels of accessibility, whereas if you're homozygous for Cs, you have very high levels of accessibility. So I'm showing you sort of the genome-wide Manhattan plot, right? So the higher the peak is, the more significant the association, right? So this would be testing every single variant in the genome for the accessibility of just this single peak, right? And it turns out that almost all of them end up have very strong cis effects, right? So they're very locally inherited, which is sort of what you would expect. So if we do this from a bulk scale, we get about 24-ish thousand total accessible regions where we can assign a QTM. And just to sort of give us some confidence in what we're seeing, we just checked some known examples of cis variants that have been well characterized. There's a few for flowering time, like the CCT9, VGT1. These two loci are associated with flowering time in maize. We see accessibility variation across genotypes, just a condensed view. Some other examples, so grassy tillers 1 has a locus about 10 kb upstream that affects this expression. We see lots of variation there. A more recent example is upright architecture 2, upright plane architecture 2. And we identified the same QTL, but also a new one, somewhat closer to the target gene that wasn't reported earlier. There's also eQTL data that exists in this population, expression variation that has been mapped previously. And we find that they have a very strong overlap with chromatin QTL, although it's not perfect. So not every single accessible variant is going to affect expression of a nearby gene. But when they do overlap, we can compare the effects of those alleles. And by and large, if we look at the overall effects, that most of them end up being either ablate accessibility or turn down gene expression. And this is just the alternate alleles that end up being at much lower frequency. And so when we actually look at the allele frequencies of all the QTL in both of the modalities, the very low allele frequency variants tend to have the greatest standardized effects. And I think that's been shown in other populations as well, is alleles that tend to be under perhaps more selection or stronger selection within a population tend to have outsized effects. But of course, because we did single cell, we want to look at the cell type specific effects, right? So this is the same locus I just showed you earlier, right? So if I just scroll back real quick, going back to this specific example here, just keep in mind this region. This is that bulk tissue profile, right? So this is the Manhattan plot specific to this local region. It's about a 2 kb window or 2 megabase window. But because we have the labels for individual cell types, we can ask, does this QTL manifest only in certain cellular contexts, right? So we find indeed, we can see the signal for this QTL within mesophyll cells, but it's totally absent in other contexts, right? So that means that accessibility dynamics is modulated by the local cis environment has only a very limited context in which it's able to happen. Maybe this is a slightly different way to think about it, is if we take all the accessible regions in the genome and we ask, what is the level of association with the local chromatin QTL or with the variant? What is the strongest association that we see? Indeed, we can see QTL in every single cell type context, but there's not a lot of overlap among them. In fact, only about 40% of QTL occur in more than one cellular context, and most of them end up being very cell type specific. And so just to give you an idea, the rows here are actually ordered identically. And so this is the significance of the most significant QTL with the accessibility dynamics of that same accessible region. So what I hope you can see is that we get very strong, significant effects. We can identify QTL when it's accessible kind of uniquely to that cell type. Now, this could also be a sort of power issue that we don't have enough cells in order to see things across multiple contexts. But we're able to identify the QTL that exists when those traits are present. All right. Yeah.
Can I have a question?
Yeah.
Do you see any example of one of these accessibility QTLs where the domain changes between genotypes? You know what I mean?
Domain
Like
changes.
you get a QTL,
Mhm.
right, on one tissue type that is unique to certain genotypes. That you see the same accessibility regions being overrepresented in a different tissue typing of gene.
Oh, I see. No, we don't see examples like that. Well, there are, I shouldn't say we don't. It does happen. So, for example, there's some number of QTL that you see in multiple contexts. But are they the same genotypes? No, we don't know that. If it's the same, yeah. Because I guess that could be an indication of like LD or long range LD or something like that. Yeah, no, it's worth looking at. But I think for me, the main takeaway is that most of the chromatin accessibility effects that we do see tend to be very specific to the cellular context in which that molecular phenotype is present. All right, and so we can also, we got like 4 million QTL. Of course, that's not very informative because not every SNP is actually independent of each other. And so what we tried to do is to find map those cis variants and try to identify something that's putatively causal using some statistical and wavy fine mapping approaches. We're still evaluating, but something that was really exciting to me is that when we look at where those fine map QTL occur, they're overrepresented physically within the coordinates of the accessible region, as well as transcription factor footprints. And so with the TAG-seq data, what you can do is you can actually look at which sequences within a globally accessible region or within a larger accessible region of the genome are protected from integrations. And those are known as transcription factor footprints or footprints that are protected from the TN5 integration. So we can identify different footprints again in the different cellular contexts, just given the integration profiles of TN5. And we see a lot of transcription factor footprints that reflect the known cell identities. So for example, DOS transcription factors in vasculature. In dividing cells, we have E2, F2, that the abaxial surface bundle sheath has canadin. So we identify a lot of the known transcription factor dynamics among different cell contexts that are reflected by the TF. And I thought what was even more exciting to me was that we can identify or cluster genotypes by their SNP profiles within an accessible region and then map out the haplotype effects, right? So if we identify the two most common groups of haplotypes, can we merge all those genotypes together and look at sort of cell type specific, allele specific dynamics? And so we identified some examples like this where you get sort of an expansion of an accessible region to include more transcription factor footprints that wouldn't have been there otherwise. So the last thing that we did was to just look at sort of how well the cell type specificity maps up between, fine mapped chromatin QTL as well as the cell type specificity of that accessible region in general. We see a pretty nice coincidence between those two data sets. All right, and sort of the last thing I just want to talk about here is that we can also just do some more basic questions about how cell type specificity is encoded within accessible regions. One of the things that I noticed was very striking to me is that if we look at where cell type specific transcription factor binding sites are present within an ACR, within the coordinates of an accessible region, they tend to be located at the very peripheries, overrepresented at the peripheries of the accessible regions. And then if we look at where the variance occurs among cell types in accessibility, roughly 80% of all the variance is happening on the peripheries of an accessible region. So this would be, each line is a different ACR, and the darker the color this is, the more variance that we are observing between different cell types in the level of accessibility. So what this shows is that most of the accessibility tends to occur around the edges of ACRs, and that's where the cell type specific transcription factor footprints are typically located. All right, and then the last question for this particular study was, well, are there specific transcription factors that have a bigger impact on whether or not, if we see variance in a transcription factor binding site, do some have a bigger effect on the overall accessibility compared to others? And when we look at that, basically the top 20 lists are all from the same, they're all from the same family, the TCP family. And data that I didn't actually show you is that they tend to be enriched in the centers of ACRs, sort of regardless of the cell type context. So this is really hand wavy, but I think that these transcription factors are playing some role in either maintaining or establishing accessibility sort of generally in plant genomes, or at least in the genome of maize. But definitely lots more to check there. All right, and just with the last few minutes, I want to talk about some of the ways we're trying to democratize the use of single cell methods and make them more accessible to many labs that maybe don't have access to some of the commercial reagents and commercial kits that we're very fortunate to have. And so a lot of this work was actually done in collaboration with Xiaoyu Tu from Celine Zong's lab. She's actually since gone on to start her own group. But the way it works is actually very, very simple in that we will isolate nuclei just like we would for a regular microfluidics-based single cell approach. We'll distribute nuclei, roughly 5,000 nuclei per well on a 96 well plate. And then we'll use uniquely indexed TN5. So these are the same enzyme, but they have slightly different adapters depending on which column and which row you're a part of. Right? And so that the idea is that each well will get a completely unique combination of TN5 adapters. And then we'll do the tag mutation per well, pull all the nuclei together, and then redistribute. Right? So we're randomizing the association, right, of these different combinations by pooling and then redistributing. And then we'll use new bar-coded I7 and I5 adapters to amplify the library, right? And so when we use this method, you can get about 1,500 nuclei per plate. So you got to do quite a few plates if you want higher throughput using this approach. But I'll just point you to the last two columns. This is with the combinatorial indexing method, which I didn't tell you that's the name of it, but it's combinatorial indexing as opposed to microfluidic indexing. And so we're able to get a much higher signal to noise ratio as compared to the commercial single cell methods. And it's at a much lower cost. You don't need your own, you don't have to buy proprietary equipment or spend money on those kits. You just need 96 well plates and the ability to order oligos. And you can do this method in your own lab. But I think what really takes it to the next level is that we can actually combine those two methods. And so, just like I told you, we can pool all these nuclei. We'll put 20,000 nuclei onto a single well and do the combinatorial barcoded pre-indexing, and then load those nuclei, massively overload them onto the microfluidics chip in an attempt to get roughly 10 or 20 nuclei per droplet, which you don't want to do at all with the normal reagents. But because we've indexed nuclei ahead of time, the chance of getting 2 nuclei with the same exact barcode combination is very low, right? And so if we aim to load between 4 and 6 nuclei onto the microfluidics chip, then we're able to get a much higher level of throughput using this approach. And so to test this, we used 7 different genotypes, 2 replicates of one genotype, where we know the genotype of these different combinations, right? And so if we are to extract those specific barcode combinations, we would know that everything that had this tag should have been genotype 1. Everything that had this tag should have been genotype 2, and so on and so forth. And so sort of overall, we get about 80-ish% of those barcodes matching exactly to the genotype that you would predict based off of the SNP level genotyping of those data. And of course, you get very nice, good quality single cell taxi data. And of course, many, many more cells, and you just only have to do a single run, as opposed to get the same throughput and cell counts using this approach, you would have to have done at least 8 times as many libraries. So we tested a few different ways, but sort of the average is around a 20-fold increase in nuclei that meet the QC standards of what you would expect with single cell data. All right, so just to kind of wrap it all up, I hope I was able to show you that you can use single cell methods to find new biology, new cell type specific biology, that a lot of the trait variation, at least the molecular trait variation, occurs within sort of context specific, in a context specific way, that we see a lot of regulatory sequence variation within maize, both at the sequence level, as well as like whether or not these genotypes are actually accessible, despite maybe having or not having the underlying sequences. That we see, that we're able to basically deconvolute the cellular context of these accessible regions and where these variants are actually having effects. And then lastly, I just quickly touched on sort of ways that we can democratize the use of these methods and just make them more, readily adoptable to any sort of molecular biology lab. And then I guess with that, I need to thank, of course, my PI, Bob Schmitz, for letting me get away with a lot of these projects. Xuan Zhang for a lot of help in library preps, our collaborators at Rutgers, Longlian Chen and Andrea Galvoti, Celine Zong and Xiao Yu at the Chinese University of Hong Kong, of course, my funding. And this is a picture of my lab. Hopefully next time I'll actually get to talk about their contributions. But with that, I'll take any more questions.